If you're like me, you read in physiology textbooks that ventricular fibrillation is maintained by something called wandering wavelets. And the idea is, this is a simulation. This is the anterior part of the heart, posterior epicardium. And it's showing wavefronts of activation wavefronts in red. And you see that they are constantly moving, they are breaking up, they are constantly re-entering, meaning just as soon as the cells have recovered, the activation front comes through and excites them again. So this is the wandering wavelet idea. Also here, constantly changing small wavefronts that form unstable reentry in the working myocardium. Well, how do we know this? We know it in the last 20 years or so, I won't go back to Gordon Moe, that we got where we had enough amplifiers that we could map out activation fronts by recording from hundreds of electrodes in the heart. And some of the data I'm going to show you comes from there. Another way is there are fluorescent dyes who, when the transmembrane potential changes as the activation front goes by, the fluorescence changes. And so you can map out the spread of activation that way. And you're going to hear more about that by Jack Rogers and Vladimir Fast later on today. Well, it turns out that VF is not maintained by one mechanism. Peng-Sheng Chen, by performing optical recordings, found that there is another mechanism where the activation is more organized. Now, it's not totally organized, in which there is a single rotor located here, meaning that the reentry is going around and around and around. And that it's fairly stable, maybe it drifts a little bit, but the rotor goes, here you see it, here it is later, here it is here, going around. And then when it gets to the back part of the heart, in this case, it's not exactly the same, and sometimes it blocks one place and sometimes it blocks another. So if you got a body surface ECG, it's going to look like ventricular fibrillation, probably coarse fibrillation, but there is still a pretty good degree of organization. So we've called this type 2 VF maintenance, the mother rotor, moderately repeatable large wavefronts that form stable reentry in the working myocardium. Well, after we came here to UAB, we found that there's a third kind of ventricular fibrillation maintenance as VF goes on, not right at the beginning. By the way, when I say we, those of you who know me know I didn't do any of this work. This work was performed by cardiology fellows, like I'm going to show you some data from Paul Tabarot and Scott Allison and Robbie Robichaud. It was performed by postdoctoral fellows. I'm going to show you some work, I think, from Nippon Chatapakorn and Derek Dosdell, and it was performed by faculty members Greg Walcott and Jian Wang. So this we, keep in mind, it's somebody else who did it, but I was part of the group. In one of these studies, we used plunge needles. So this is a needle that we're going to put through the wall of the ventricle that had 6 electrodes on it, with 6 towards the endocardium, 1 towards the, epicardium. They're coming in from the epicardium. This is endocardium. They're 2 millimeters apart. And we put these needles into pigs in some studies and dog in another. And we would electrically induce VF and we would just record for 10 minutes or until the VF stopped. And so here's the results from typical from the pig. When the activation front goes by in these unipolar recordings, there will be a rapid downslope in the signal. So you see here activation fronts are going by. This is 1 second of data, so it's activating about 10 times a second. And about the same rate all the way transmurally from the endocardium to the epicardium. Same thing in the dog, it's going a little bit faster, but you have basically the same rate from the endocardium to the epicardium for the first minute of VF. But then as VF continues, we noticed something fascinating. In the pig, the VF slows a little bit, but it's still going about as fast on the endocardium as the epicardium. But in the dog, it's going faster, still fast on the endocardium, but it's going more slowly on the epicardium. And we could track out some of these activation fronts. This looks like it's starting here, going out through the wall. This next one is going about halfway through the wall and blocks. The next one's going through the wall. So it looked like that the wave fronts were originating from the endocardium as the heart was getting ischemic. We didn't perfuse, we just let the heart go, and so it was getting more ischemic. Well, why should that be? Well, partially because you've got oxygenated blood in the LV, so maybe the endocardium is not as hypoxic, but also there's a specialized conduction system. And occasionally on these plunge needles, we would see these little spikes. Can you see them? And so we thought maybe that those were Purkinje spikes. Well, what's the evidence for that? Well, it's obvious. There's arrows pointing at them. I'll give you some more evidence for that in a minute. So we knew that the distribution of the specialized conduction system is different in different species. And it's different in ungulates, pigs, sheep, than it is in dogs and humans. In all of them, there is a layer of Purkinje fibers on the endocardium. And that's stained, the glycogen is stained black here. This is sheep. So that's true in all the mammalian species that I know about. However, in dogs and people, all of the Purkinje system, or 99% of it, is there, right there at the endocardium. But in ungulates, sheep and pigs, not only do you have the Purkinje fibers along the endocardium, but bundles traverse the wall. Don't ask me why. It traverse the wall and go all the way out. Here's the epicardial vessel. They go all the way out to, there's 200 microns, almost to the epicardium. So maybe that was the reason that the pig kept going at the same rate transmurally while the dog didn't. But perhaps these activations are arising in the Purkinje system. So we wanted to see, could we map out activation on the endocardium from the Purkinje fibers? The Purkinje system, this is a dog, you can see it here. This is at the end of a study where Lugol solution was put on, which stains the glycogen of the Purkinje fibers purple. So there is this pattern on the endocardium, and we tried to map it. Well, how did we get to it? We took the heart out of the animal. These are dogs. Cut through the RV, the septum, exposing the LV. We put a plaque. Do you see those 4 sutures? We put a plaque there with 500 electrodes on it, and we would record, we'd induce VF and record till a VF stopped. You can see, in this case, it's right here at the base of this anterolateral papillary muscle. So we would record. So the first question is, did we see what we thought were Purkinje fiber activation? And yes, we did. This is where this represents the 500 electrodes. This is a bunch of static maps 2 milliseconds apart. And these are 5 electrodes, and their location are shown here, A, B, C, D, E, F someplace, F. We saw these rapid downslopes shown with the red arrows that we thought that was the working myocardium activating. But then we frequently saw, because we're now right on the endocardium, it's not like a plunge needle that could be 2 millimeters away with that electrode. We frequently saw these activations preceding or coming after the activation that we're showing in yellow. This is the first temporal derivative, which makes the thing stand out better. So we mapped out these activation sequences that we thought were Purkinje in yellow and the working myocardium in red, So we saw activations arise in the working myocardium and go into the Purkinje system. We saw activations arise in the Purkinje system and go into the working myocardium. And then we also saw something fascinating. Nothing. Sometimes we wouldn't see anything for milliseconds, and then something would just show up in the Purkinje fibers, de novo, and then did you see it get into the working myocardium? So there were activations that looked like they were not reentry, but arising in the specialized conduction system. So what we wanted to do next is, well, we thought those were Purkinje. We did the same study, except before we did put the plaque on and induced VF, we swabbed the endocardium with Lugol's solution, which kills the Purkinje system. Then as a control, we swabbed saline. And so 6 hearts each, we induced VF and we recorded. The top recording is from the control with saline. You still see these spikes that we think are Purkinje. Here's the temporal derivative. Here is the one with Lugol's, and it looks totally different. Also note that they stop spontaneously. Well, I mean, the VF's got to stop sometime, but they stop spontaneously. So when we looked, here's what we saw. What we also did is we put a few plunge needles around the edge of that plaque, and here is one. So here's a plunge needle now with epicardium on the top, endocardium on the bottom, 6 electrodes again. And every 2 minutes, we see the same thing. The endocardium, this is the control. The endocardium keeps activating rapidly, slower activation propagating out to the epicardium. With Lugol's, we didn't see that. We saw slowing activation that looked like it was going transmurally. And also notice that this VF stopped spontaneously between 4 and 6 minutes, while in the control it went on for between 8 and 10 minutes. And we saw that in all of the animals. The 6 animals that had Lugol's applied to the endocardium, this is the activation rate. It slowed, and then when it got down to about 1 activation per second or 2, it stopped with a mean of at about 5 minutes. The saline controls, well, they slowed down too, but it took longer. And again, it stopped when they get down to 1 or 2 hertz at about 9 minutes, and that time is significantly different. So here we are talking about a time not important for the ICD, but important for external defibrillation and resuscitation, where it looks like the Purkinje system is important in maintaining the ventricular fibrillation. We gambled on something. It looked like, as you saw with that plaque, it looks like there were wavefronts that we could map out. So we wanted to do something a little more natural than an isolated heart sliced open. So we gambled that we could take a 64 electrode basket that's was used clinically, and perhaps we could map out VF with it. So here's a study in which there were 2 baskets, one in the RV, one in the LV. You make out there's 8 struts. There are 8 struts. Each strut has 8 electrodes on it. And we induced VF and let it go for at least 7 minutes. Here's in early VF. This is the 64 electrodes recordings from the LV basket. There's an 8 along one strut, there, 8. I think it's going, in endo to epi, I think. Here's 1 electrode from the RV, and here's limb lead 2. So there's VF, and that's what you expect. But this is not what, we did not expect this. Sometimes after VF had been going on for about a minute, the endocardial activation sequence totally changed. It's small, but can you make it out? There's 1 big activation going very fast, and then there's nothing. There's no sign of reentry. Then another big activation, different coupling intervals. The RV is not doing that, and the limb lead 2 still looks like VF. Well, we mapped those out, and here's an example. This is 1 second of data. Here are the 64 electrodes from the LV, 8, 8, 8 each strut. Here's the RV. Every time we thought there was a muscle activation, which we're showing here with the red, we have a red bar, and every time Purkinje, which is green, we put a little green bar. Notice that the 2 ventricles are dissociated. The RV is going twice as fast as the LV. Here, these represent the activation maps of these 5 activations during this 1 second. This is kind of like a spider web. Imagine that we took those 8 struts of the basket and we opened it up. So you have the apex in the center, and then you have these 8 spots going out. So that represents the 64 electrodes. The activation times are shown in color, which the earliest activation is in blue, and the latest activation, which is about 40 milliseconds later, is in red. And here are those 5 cycles. Here's the first one. Where do we see earliest activation? In the Purkinje system, over here at the base, which then got into the working myocardium and then propagated around. So latest activation is on the opposite side of the heart from earliest. No indication of reentry at all. Here's the next cycle where the earliest activation was in the Purkinje, and then back into the working myocardium and late over here. Here's another one. So different activation sequences here. Beat 4 is earliest over at the base of the Purkinje here, with latest activation on the opposite side, and the same thing for the next beat. So they look like foci arising in the Purkinje system. Well, what could that be? It could be abnormal automaticity, but it also could be triggered activity, of which I know it's much more complicated now, but back in the old days when it was easier, there were early after depolarizations and delayed after depolarizations. There are drugs that prevent these, but they're not clean. They do lots of other things. But I'll show you the results here with 2 of those drugs, flunarizine and pinacidil. Here's 8 animals with the control where we didn't give any drug, and we saw that synchronous pattern about 1/4 of the cycles between 2 and 7 minutes of VF. A very important time for resuscitation, not for ICDs, but for sudden cardiac arrest. When we gave the pinacidil, that among other things blocks EADs, we never saw the pattern. Then when we gave flunarizine, which blocks DADs, it had no significant effect. So this has led us to say that there is yet a 3rd kind of VF, which we call ventricular electrical synchrony, almost synchronous activation of the Purkinje fibers, probably arising from EADs, that spreads into the subendocardial working myocardium and propagates towards the epicardium with variable conduction block en route. Okay. But notice that pattern is there 1/4 of the time. What's there the other 3/4 of the time? Reentry around the apex in almost all cases, not coming from the Purkinje. It does in a few cases. But what we would see is here's 4 consecutive cycles. Each cycle, earliest is blue and latest is red. So here, earliest activation is here at the apex, spreads out as it goes around the apex. It's spreading out up to the AV groove. And notice the latest is right next to earliest, because I'm going to jump now to the next cycle. So this went around and is spiraling out, gets to here, re-enters and goes around. There's beat 2, here's beat 3, here's beat 4. So we have a mother rotor at the apex most of the time between 2 and 7 minutes of VF in dogs and pigs. in this case also. Why? What's special about the apex? Well, I don't know, but there is one thing special about it, and that's the orientation of the muscle fibers. We're looking up at the apex, the endocardium, of the epicardium. The heart's been boiled. Here's RV, LV, here's anterior interventricular groove. The fibers, I don't know whether you want to say they're spiraling in or they're spiraling out, but perhaps it could be circumstantial, but perhaps something about the special fiber orientation at the apex is responsible for that. Okay, I started late, so I'm going to change gears here for defibrillation. I'm 80% retired. This, in all likelihood, is going to be the last talk I ever give. That's so sobering, I have to have a drink of water here. I thought I would frame the discussion for defibrillation with some words of wisdom that I have gathered that are not the things I learned about the scientific method as a doctoral student. So as I said, I'm about 80% retired. Why did I do that? It was no one thing. But when I moved here in '94, I was 52 years old. I felt I was at the peak of my powers. I thought I was riding a white horse. I was doing good. I'm trying to understand things about basic science, to have a good time, but also to help people. And it went along fine. But then one day I looked in the mirror. That's still Clayton, whatever his name was, Moore, the Superman. And time had kind of caught up with me, and I thought maybe I better get off the white horse before I fell off of it. So you might say, well, gosh, what am I doing now with all of that time? Well, I have picked up some new interests that are keeping me occupied. So let me give you the words of wisdom. The first observation. The first observation is that a wrong hypothesis can be very useful. And I'll give you an example of a combination. Up until 20 years ago, defibrillators delivered what was called a monophasic shock. You'd charge up a capacitor and it would passively discharge through the leads. Janice Jones had come up with, and some others, with the idea of a biphasic waveform in which, while the capacitor is discharging, you suddenly change the polarities. You switch it and change the polarities. So if this is positive, this is negative. Now, Janice Jones had a hypothesis on how this would work, why that should defibrillate with lower energy and her idea was this. We knew that during VF, the cells are activating typically very quickly just after they have recovered enough. They're still in a relative refractory period. So their transmembrane potential is elevated. So we know that there's some parts of the heart where when you give a shock in the extracellular space, it would depolarize hyperpolarize, don't let me get this backwards. It would hyperpolarize the transmembrane potential. So that she thought then if you then, in that same part of the heart, reverse the polarity, it would depolarize, and it would be easier to depolarize the tissue because by pushing the potential back down, you made the cells more responsive, so they'd be easier to activate. So she thought that the purpose of the first phase was to bring the membrane potential back down where the second phase could excite the tissue more easily. Well, if that's true from her hypothesis, I hypothesized then, okay, you would think that the important phase is the second phase. It's the one doing the depolarization and stopping the wavefronts. So you would think, the hypothesis was, you needed more charge in the second phase than the first phase. Well, we tested this. we used a monophasic waveform that was 10 milliseconds long. Then we kept the 10 millisecond total duration of the waveform, but we had we had a 2.5 millisecond first phase and a 7.5 millisecond second phase. I'm going on out 5 milliseconds each, all the way out where the first phase was 7.5 milliseconds, and just a little bit of charge in the second phase, 2.5 milliseconds. And the results were just the opposite of what my hypothesis would predict. Here's the DFT. So the DFT is the shock strength which you need to roughly depolarize about half the time. Here's voltage. These are some really big electrodes on the epicardium, so these are some really low DFTs. About 150 volts and about 3 joules. Notice the waveforms that my hypothesis said were going to be the best were the worst. They were much worse than the monophasic waveform. The really good ones were over here that had 3/4 of the charge in the first phase. This study was done, I don't know, 20, 30 years ago, and what's nice about this is now almost every external and internal defibrillator uses a waveform similar to that. So my second words of wisdom, I know this goes against scientific method, is you can improve something without understanding how it works. Just brute force, just try to optimize things. Of course, I'm enough of a basic scientist, we want to know how things work, because if we knew the method, the mechanism, then maybe we could come up with a better way to defibrillate. So we have done a number of studies trying to determine the mechanism of defibrillation. What we found early on was it was thought about 1975 or so, from a study from Doug Zipes, it was thought that to defibrillate, all a waveform a shock had to do was halt the activation wavefronts during VF. And if you halted all those activation fronts, then the VF would stop. Well, we found out that no, you had to do more than that. That you could halt the wavefronts of VF, yet the shock itself would engender new activation fronts that would throw the heart right back into VF. And that took a bigger so if you then kept giving higher and higher shocks, then you might still generate a new waveform, but it wouldn't go back into VF. So the rate limiting thing on why you need a certain shock strength to defibrillate is so you don't reinitiate VF. So people tried to say, well, okay, how does a shock reinitiate VF? This is a study of optical mapping performed by Igor Efimov, not our group, in which he had isolated rabbit hearts and he performed optical mapping here, the anterior surface. Again, here's the anterior interventricular groove, so here's RV, epicardium, anterior LV. Most of his shocks were not high strength, and they were not given during VF. But how he found the VF was initiated was by this mechanism. So with the optical recording, he could record a signal proportional to the transmembrane potential. So he found in this case, after the shock, that he had this area that was depolarized, and then a rapid change where right next to it was an area that was hyperpolarized. And then he could follow the activation fronts after the shock. And what he saw, and notice that you don't have that rapid change up here. Here's where the rapid change is, right here. Well, what he saw was an activation front launched from there immediately after the shock, 5 milliseconds after the shock, that went around and formed reentry around this area, and then went back and degenerated into VF. So according to this mechanism of defibrillation, the way a shock reinitiates VF is by immediately launching a wavefront that forms reentry. Okay, good. However, there's some catches here. Like, for example, the shocks were not always up at DFT strength. It turns out on the epicardium there are not many collagenous septi the way there are through the wall. So when you go through the wall, according to the New Zealand group, you don't have these very large areas of hyperpolarization and depolarization. You have much smaller areas. We did a number of studies where it didn't look to us at all like this is the mechanism for defibrillation. I'll only show one because it's a clinical. This is a study Greg Walcott did with Vance, Plum, and was Neil involved in Neil K, which during ICD implantation, there's a shocking electrode. They put an electrode into the coronary sinus with, I think it had 18 electrodes on it, and an electrode in the LV that had 36 electrodes on it, and they recorded. Here's what they recorded after a shock just a little bit below the DFT, and the shock failed. Notice these hash marks are 100 milliseconds apart. So they didn't see anything for like 200 milliseconds. And then they saw a wavefront come, this happens to be half of the LV catheter. Again, from the apex going out, here goes another wavefront out, and they come faster and it goes back into VF. Here's a shock, the next shock strength up, and it succeeded. They still had a fast activation, but again, there's no sign, there was no sign of reentry there. Nothing showed up immediately after the shock. But again, this is only 36 electrodes. It could have been somewhere else and missed it. So we did a study, you remember the plunge needles, where we put plunge needles all over the RV and LV. And here's a representation. It's 3 different views. Let's just look at this anterior view. Here's a plunge needle with the 6 electrodes from epicardium to endocardium. Here's a plunge. Here's a plunge. There's a lot of them down here. And we gave shocks that were very close to the DFT, and some succeeded and some failed. And so I'll show you a typical example of a shock that failed, but just were very close to the DFT. What did we see right after the shock? Nothing. 62 milliseconds later, we see activation show up towards the endocardium that then spreads out like a focus. These are every 10 milliseconds. So it didn't look anything like the reentry immediately arising after the shock. It looks like a focus coming from the endocardium. However, it's not completely clear-cut, but generally, flunarizine and pinacidil did not affect the DFT. So maybe this is not automaticity or from the Purkinje system. And another piece of evidence against that is a study that Peng Sheng Chen did. This is an isolated rabbit heart. He froze the inside, but the heart was in a warm saline, so about a millimeter of myocardium survived on the epicardium. It stained red with the supravital dye. And he performed optical recordings of shocks close to the DFT. And here are the activation times. Here's 4 hearts. There again was just what we saw, but not on the endocardium, and couldn't be involving the Purkinje system. But here the first activation showed up at 52 milliseconds, this one at 36, 69, 54. He could also record with a different dye intracellular calcium levels, and what they noticed is that the calcium went down earlier after the shock in the area in which the activation later occurred. So I don't think we still know exactly the mechanism of defibrillation, even though it's done all the time. Also, it may not be one mechanism. Here's a study where we recorded with plunge needles with VF lasting 10 seconds and we give a shock. And here you see an activation about 30, 40 milliseconds after that. But here, with 5 minutes of VF, We don't see anything for 100, 150 milliseconds, and we frequently see a Purkinje spike in front of it. So maybe the mechanism of, so we've seen that the mechanism of VF is not one thing, and it can be different during different durations of VF. Maybe the mechanism of defibrillation isn't one thing either. I have two more. Yeah, we're okay. You should always question your assumptions. Sometimes you make an assumption and you don't realize you're making the assumption. We know from theoretical and experimental studies that there are waveforms that are more efficient for defibrillating than a truncated exponential. I mean, you got the truncated exponential because you charge a capacitor and it discharges. It turns out that waveforms in the first phase that do the opposite of that, start at 0 and increase, defibrillate with lower energy. Not voltage necessarily, but energy. This was a moot point because you couldn't really make that waveform with an implantable device. It's of a little more importance now because there have been some developments in electronics, and it probably now is possible to make that waveform in both an external defibrillator and an ICD. So there have been a couple of studies that the ascending ramp first phase had a lower DFT energy, but then the question we asked are, well, does that mean the shock causes less damage? And I'll show you evidence. Shocks do cause some damage. We can discuss how important it is, but they do cause some damage as judged by troponin release and later on by histologic necrosis and things. So I was wondering, what is it that causes the damage? Is it and I thought, well, it's the strength of the shock. Obviously, I assumed that. And then the question was, well, is it the peak voltage or is it the energy? And so we did a study to try to determine that. So we did a study where we looked at 3 different ascending waveforms. One was 4 millisecond long, one was 8 milliseconds long, first phase, 16. They all had a second phase, reverse polarity, that was half the amplitude of this peak voltage and lasted 3.5 milliseconds. I'm going to show them superimposed here because for space reasons in this next plot. We also looked at the 16, 8, and 4 millisecond descending biphasic waveforms. We looked at 16, 8, and 4 millisecond square wave, rectilinear, because this, in theory, is the waveform that defibrillates with the lowest voltage. Here's a Russian waveform, the Gurvich, which is a form of biphasic, and here's our control, our truncated exponential biphasic. This is a typical waveform for an ICD. We determined DFTs in 6 animals for these waveforms, because then we're going to pick a couple and do a study to look at damage with troponin. And it worked out pretty well. The green are the ascending waveforms, and they were low energy, although this 4 millisecond descending was low energy too. But the 8 millisecond ascending ramp was a low energy, so we chose it to study in our damage protocol, which is protocol 2. We then chose a low voltage waveform, which is the 8 millisecond rectilinear, so we used it too in the study. And then we had our truncated exponential as the control waveform. All right, so what we wanted to do is give a shock that was pretty sure to defibrillate and then measure troponins at baseline and every 2 hours after that, troponin I. So what shock strength should we use? Well, normally, do you still use a 10? You don't even determine DFTs anymore, do you? But it used to do you'd find the DFT and you'd take a 10 joule safety factor. So that's what we decided to do. Notice that the control is 20 joules, so we decided to use 30 joule shocks. The question is, what should we do with the good one, which is the ascending ramp, which was only a 15 joule DFT, should we use 10 joules above that 25 joules, or should we make them all the same at 30 joules? And for some extraneous equipment reasons, we wound up making them all the same. And I'm so lucky that that happened, and you'll see why. So then you had to have animals, so the shock, we gave the shocks all 3 shocks were given at 30 joules, which means then, of course, that the voltage, peak voltage is different. Here's the rectilinear with the low peak voltage. It turns out this ascending ramp actually has a significantly higher peak voltage even than the truncated exponential. So then we wanted to look and see, so what's going to be important here? Is it going to be the energy, or is it going to be the voltage that determines damage? Well, here's the truncated exponential. Here's the troponin eye level. Baseline got up to about 2 nanograms per milliliter at 4 to 6 hours, and then started coming back down. Here is the ascending ramp. Even though it's the same energy and a higher voltage, it caused half of the troponin release. About 1 nanogram per milliliter. And then here's the rectilinear, and it actually caused significantly less than the truncated exponential, but significantly more than the ascending waveform. So sometimes you get lucky. Had we done that study with this waveform at 25 joules instead of 30, then on my assumption, I'd have said, well, obviously it's the energy that's important, because this was the lowest energy one and it caused the less damage. But we got lucky and we tested them all at the very same energy. So this is the one with the highest voltage and the same energy, and it causes the least damage. So my assumption that the big important thing that causes cell damage from shocks is the shock strength isn't right. The shape of the waveforms looks like it's equally important. Here's my last one. As you get, good. You know, you get a lab that's pretty big, there's a lot of people in it, you're traveling, you're going to meetings, you're writing grants, you're reviewing manuscripts. You got to be careful to know what's going on around you and what's happening when you're not there. So you need to be aware and watch your back. Now this is the obvious one, but this is a roller coaster in Copenhagen. Who wears a coat and tie on a roller coaster? I'm worried about that guy behind my back too. Thank you very much.
Okay, does anybody have any questions for Dr. Ideker? All right, great. Well, Ray, you know, you're ready to be done?
No, go ahead.
I know that you had worked with Michelle or spent some time in Bordeaux looking at the cardio insight system and atrial fibrillation.
I visited there.
Oh, okay. Well,
I visited there. I mean, who wouldn't? I was there last week. I got invited to give a talk or two there, and of course I went. But no, I've not worked with him. Igor Efimov has.
Yeah. I guess I was interested in what your thoughts were on that system and atrial fibrillation, and if there was any hope for those of us who toil away in the EP lab trying to treat atrial fibrillation.
You're talking about the very low, where you give a series of low voltage shocks and try to defibrillate.
It's the surface mapping system that you have
no.
to solve the inverse
About that
solution.
defibrillation system. No, I don't know much about it. Yeah.
So, okay, maybe a broader question. Is there any hope for those of us who are trying to treat atrial fibrillation? From a mapping standpoint, will we ever be able to map AFib?
Well, you're keeping up with the mapping more than I am about trying to locate rotors and ablating just those sites and so forth. The other thing I was talking about, but I haven't seen anything come out of it lately, was Igor had these methods where he was just going to give some pretty low-level shocks. I mean, I don't see how you ever do that for VF, because what if it didn't work? For AF, if it doesn't work, you could try it 5 times and you haven't lost much. But I'm hopeful, but I haven't seen anything come out of that more recently.
Yes.
Hi, Guy.
Hi, Ray. This is a great talk, and it's just delightful to hear you present. We've been also interested in VF and the dynamics of VF and how it progresses in time. And one of the ways we've approached it is to look at extracellular potassium. And to look at VF through extracellular potassium concentration. So if you pace faster and faster, the release of potassium from the heart doesn't exceeds that of the reentry and the reversal. So you have narrow clefts between cells, and you also have T-tubules where it's been supposed and theorized that extracellular potassium accumulates. And that has an impact on essentially your resting potential and your ability to defibrillate. And so with time, potassium exceeds 20 millimolar in concentration, and then you can't defibrillate. It's simply too depolarized to reactivate any of the channels. And so the interesting things that you showed here in terms of 7 minutes has a lot to do with this accumulation of potassium. And then the Purkinjes, however, are more resistant to it. Okay. And that is it lines up beautifully with this effect of potassium. Differences in the T-tubule morphology, and also diffusion of the potassium into the endocardial space, as opposed to the middle of the tissue where the accumulation is likely to be more severe. So it's a wonderful.
Are these isolated hearts?
So this was done with isolated hearts where we controlled flow.
So can you control the extracellular potassium?
Yes.
And what?
Right.
Have you looked
so
at that?
if you perfuse with crystalline solution during the VF, you slow it down. But if you slow down flow, as you would have in an intact animal, because the fibrillating heart is actually, it has an impact on flow and perfusion of the heart itself. Right. And that means that potassium accumulates faster. Okay. And so this duration of how VF is altered by potassium is time dependent and flow dependent.
Right. I mean, there's so many things going on. For example, Greg looked at, and we've seen public, you can look with ultrasound or fluoro, and during early VF, the heart is hypercontracted.
Right.
But then as we get into these time periods, it's relaxing and dilating.
Right, so the dynamics are quite complicated. They depend on the severity of ischemia, of course, but also flow. And past a certain point, potassium is too high and you can't defibrillate. It's simply too high.
Yeah, that's the trouble with the world. It's also complicated. I mean, not that I'm fixed on wine or anything, but you read about what yeasts are going to ferment the wine. It's not one. I mean, even if it starts off as one, is the alcohol levels going up during maceration as that goes on? The relative amounts of different yeast change, and so nothing's
Yes,
pure.
ultimately wine is the answer, yes.
Yes.
More words of wisdom, wine's the answer. I think I propose some more words of wisdom as regards to you. Truth and wisdom prevails and endures. I don't know if people in the audience are all aware of it. These are studies that date back decades, and the wisdom and the truth of it does endure and contributes ongoing. It's not an end process. Congratulations, Ray.
Oh, thank you very much, Vance.